This video is B1, cell biology in a nutshell. So it's everything you need to know for the topic B1. I've left the triple content at the end, so if you're double you can just watch up to that point, although we have seen that content come up on the double papers even though it's described as a triple practical. We've got 3 required practicals in this topic and we'll go through those as we go through the topic. So we start off with eukaryotic and prokaryotic cells. Now, eukaryotic, think you and I, we're animals, and also the plants go into that category as well. Now we need to know this because if we have a question that says, name something that's in, name a subcellular structure that's in a eukaryotic cell and not in a prokaryotic cell, and we've forgotten what those words represent, then we can't answer that question. So the eukaryotes are the animal and plant cells, and the prokaryotes are the bacteria. Now, we are not going to be expected to draw a bacteria, but we are, or bacterium I should say, but we are going to be expected to draw the animal and plant cells. Animal cells, really simplistic. 3 key points to look at there. We've got the nucleus containing all of the genetic information. We've got the cytoplasm where chemical reactions take place, and we'll look at more of that on the next slide. And we've got the cell and membrane, which controls what enters and leaves the cell. If you think about your skin cells and the flexibility, you can push on your skin and it doesn't damage the cells. That cell membrane is very flexible. They've got to kind of think of animal cells as kind of like balloons, okay, very flexible. If you take a plant leaf and you bend it, you see the damage that you cause. It kind of breaks the cell wall that the plant cell has. So plant cells got those 3 key points again, those 3 subcellular structures. In addition to that, it's got the chloroplasts where photosynthesis takes place. It's got the vacuole containing the cell sap and also the cell wall. And think of it like the walls of a building. It's on the very outside of the cell, and that gives it structure and support. It's made of cellulose. So we may have to draw the animal or the plant cell. Then we've got our prokaryotic cells, the bacteria. They're smaller. They're actually smaller than the mitochondria, which we'll talk about in a bit. And so that's why mitochondria actually wouldn't be able to fit in a bacteria. Popular question with the exam board, they like to talk about that. It's a single-celled organism, doesn't contain a nucleus. Instead, it has all of the genetic information as a single DNA loop, and then these rings of plasmid, which we can't really see on this picture, but we see them when we're talking about genetic modification as kind of circular loops. No mitochondria, as I just said, because they are smaller than mitochondria, has a cell wall, and they multiply by binary fission. So later on in this topic, we'll talk about mitosis. Bacteria do not multiply by mitosis. They multiply by this process called binary fission. Could be a quick 1 marker that would be worth remembering there. So we might have a question that says, name 2 ways the bacteria is similar to a plant cell. And you could say something like it has cytoplasm and a cell wall. And it might say one way, name 1 way it's different from a plant cell. And you could talk about that flagella, okay, helps movement there. Not all bacteria have them, but in this particular picture, we can see that. Okay, moving on to the next slide. So within the cytoplasm, we've said that this is where chemical reactions take place, and it's actually within more subcellular structures. Okay, it's not just randomly happening in the cytoplasm, but within that cytoplasm we have the mitochondria, and you should remember from Key Stage 3, that's where respiration takes place. And respiration is all about releasing energy. It's about releasing the energy from that store that is glucose. We can never say produces or creates or makes energy, because that's not possible. As we learn in physics, energy cannot be created or destroyed. So it's just using the process of respiration to release the energy from the glucose. And that energy can be used for everything that's needed in the body. Then we've got the ribosomes. We don't really learn much about the ribosomes unless we're triple science and we get into paper 2, and then we finally look at protein synthesis. It's just an extravagant way really of saying making proteins. So if we essentially think of it as we eat proteins, we break them down into amino acids so that they can get into the blood, and then when they get to the ribosomes, they build back up into proteins, and those could be hormones, they could be enzymes, they could be things like collagen, growth and repair proteins, those sorts of things. But for now, we just need to know ribosomes are where protein synthesis takes place. And then the chloroplasts, which are just in the plants, and we've talked about those already, absorbing light. It's the actual pigment chlorophyll, which makes the leaves green, that absorbs the light, and that's where photosynthesis takes place. We don't see those chloroplasts in the root hair cells because they can't absorb light because they're under the ground, but we do see a lot of mitochondria in the root hair cells, and we'll come back to that later on in this video. So when we look at those original cells, we think of those as just kind of the generic cells. We're now going on to cell specialization, and the exam board have come up with the 6 cells that they would like you to know about. Now, it doesn't mean that they're not going to throw in a curveball, different type of cell, but these are the specialized cells they want you to be very familiar with, particularly for double science, triple science. You may get something a bit different where you've got to use, maybe some labels on the diagram to figure out, why has it got mitochondria or whatever it might be. So let's look at the animal cells first. We've got a sperm cell. We've got on the head of the sperm, where the nucleus is, we've got these enzymes, and they help to get into the egg. And it's actually only the head of the sperm that gets into the egg, and then the tail breaks off. In the middle section, then we've got loads of mitochondria. That's where that respiration is taking place, and that respiration is providing the energy for the movement, for that tail to move so that the sperm can get to the egg. Then we've got a muscle cell, again using lots of energy, that energy coming from respiration, so again lots of mitochondria found there. Also protein fibres for contraction of the muscles when we're lifting things or moving our arms and our legs. Then we've got a nerve cell. We come back to this one again in paper 2 when we look at neurons and the reflex arc, and all of those yellow parts are kind of like the plastic coating on a wire. They are really good at insulating the rest of the nerve cell, and it's got this electrical impulses going through those orange parts, okay? Dendrites stretching out on the left hand side there to collect lots of information, and then travelling along. It's called an axon, but you don't need to know that word, and the insulating material is called the myelin sheath. Again, you don't need to know that, but it's better than just saying along the yellow bit. So we've got electrical impulses passing through those large surface area, very long, taking information around the body. Then we go on to the specialised plant cells. So we've got the phloem, transporting all of the sugar that's made in photosynthesis to different locations. So the process of moving that sugar through the phloem is called translocation, transporting the sugar to different locations. And that picture just here is of the phloem, and there's cells stacked on top of each other. There's multi-directional flow, and the ends of the cells where they're stacked on top of each other have got perforations in, small holes to allow that sugar to easily transport through the end of each cell. And then we've got the xylem. They are, again, these kind of tubes, a bit like a straw. As soon as you drink a bit out of the straw, more water comes up, or more drink comes up through the straw. And they stretch all the way from the roots to the leaves, and as water evaporates through the leaf, through those stomata, more water is drawn up through the stem, through the xylem, from the roots, and more water will go in at the roots at that point. So it's just like a straw. Now these are also cells stacked end to end, but the walls have broken down, forming that continuous straw. And it's that stringy bit of the celery that we're talking about here, okay? So that's what those tubes are like. Just think of them like straws. As soon as any water evaporates from the leaf, which will happen more on a warmer day, more water will be drawn up through the roots and move through that xylem. You don't get air bubbles in here, okay? It's just like when you drink through a straw. And then we've got the root hair cell found in the root of the plant. You can see the bottom picture there is the root, and then you can see all the tiny root hair cells coming off of that. Again, no chloroplasts because they're underground, no need for them. They do have mitochondria, and this is for active transport, and we'll talk about that right at the end of this video, but that's movement of substances from low concentration to high concentration, which needs energy to occur. Again, you can see here this large surface area, always talking about that in biology, allows maximum absorption. It allows more water, more nutrients, more minerals to move into the root hair cell. More is a key word there. Doesn't just allow them to go in. If there was a smaller surface area, they'd still go in. But having a large surface area allows more absorption. Cell differentiation, we've got this word that is just, basically cells become different, they differentiate, and we seem to like, the exam board seem to like the word differentiate and then write and become specialised. So try and get those both into your answer. When cells differentiate, they're becoming different, they're becoming specialised. Now, we've got this idea of the sperm going into the egg, we call that fertilisation, okay, the fusion of the gametes. And then as soon as that happens, we get this mitosis, which we'll talk about in a bit, which is cell division. And at this point, the cells are really basic, okay, we can see in the embryo picture here. But cells then differentiate, they become specialised into all of the different cells that we need in the body. Examples here, nerve, muscle, skin. And that happens very quickly in animals, but at any point in plants. So we could be looking at a 100-year-old oak tree outside, and it's still got cells that have not yet differentiated. Okay, so differentiation, very quick in animals, any point in plants. We go on to our first required practical, and you can see there we've got the method of how to prepare and observe a microscope slide. Classic one for our 6 marker, particularly for the doubles, writing out methods. Try and get it down into 6 points. So this is looking at onion skin. So we take that thin layer of the onion and put it onto our microscope slide. We add the iodine solution, which stains the subcellular structures. We put a cover slip on, try and remove any air bubbles, and it gets it nice and flat, and then we're able to view it on the stage of the microscope. And we start with the lowest power. Why? Because it is easier to focus. Classic question, okay? We start with the lowest power, it is easier to focus. We use the coarse adjustment knob, and then we can fine adjust as well. Once we've done that, we can go to the next power objective lens. Okay, and we can keep going until we get to the highest power that we can clearly see. And refocusing should be really easy as we work up, if we focus correctly, on the lowest power. And the microscope stage can move up and down, okay, and that's how we focus things. So what we want to do is have it as high as possible and lower it down. If we pull it up, then the objective lens can hit the slide and it can damage the lens or damage the slide, okay? Another classic question. So we move it gently and we move the stage down so that we don't allow that to happen. We've got our magnification, size of image and real object equation there. Yes, we're going to need to learn this equation, okay? We can remember image on the top of the triangle, so they're likely to give you a picture, so the image is always on top, okay? You might have to measure something with your ruler in millimeters, don't go for centimeters. Measure it in millimeters, turn it into micrometers or micrometers by multiplying by 1,000, and then divide it by the magnification to get the real objective size. That's likely to be what they'll need you to do. Okay, we've got 2 types of microscope. We've got the cheap microscopes. I say cheap, they're a couple of £100 that we use in school. They're called light microscopes because they have a light in the base which we shine up onto the slide, which allows us to see what we're looking at. Then the good thing about these is that they're cheap, not great resolution or magnification. We can't see too much beyond the cell wall and the nucleus. But then we've got the electron microscope. It's very, very expensive, a few £1,000s. And the words we must use to describe the benefits of an electron microscope, by the way, not electronic, electron, like electrons in an atom, it can see really small things. Okay, so think of it like that. Do not call it an electronic microscope. It's an electron microscope. And the words we must use to describe its advantages: better magnification. So that means that we can zoom in more, so we can see more things, but we can't write see more things. We've got to write better magnification or increased magnification, and increased resolution. And resolution is about getting that sharp image. But again, don't write sharp image, write high resolution. Those are the words you must have: magnification, resolution. Then we might have to work out the total magnification. So we've got the eyepiece lens at the top and we've got the objective lens. And whatever those two magnifications are, we multiply them together to get the total magnification. So we've got here an example of times 10 on the eyepiece lens, times 40 on the objective lens, and multiply them together, and it's multiplied by 400 times. So we're seeing those cells 400 times bigger than they actually are. We've got, for doubles certainly, and triple foundations, we need to know that 1 millimetre is 1,000 micrometers, so we can do those conversions. And then if you're triple higher, you'll also need to know that we've got 1,000,000 nanometers in a millimetre. Okay, that one has come up on a paper recently, so that is something that you'll need to be able to work with. Standard form, 10% of this paper is going to be maths, so to try and get some maths in, we incorporate standard form. And when we're looking at really small images like this, it's a great opportunity for them to try and get that in. You must have one number in front of that decimal point. We could not say 6,027 times 10 to the 2. That's not a thing, okay? So it's 6.027 times 10 to the 5, and that means we've moved the decimal point 5 places to the left to get it into standard form. Okay, so if we go the other way, if we've got a really small number, then I don't appear to have got the full number on that bottom one. It should be 4.3 times 10 to the minus 4. I don't know why the whole number isn't there. So 0.00043 is 4.3 times 10 to the minus 4. Okay, let's move on. So when we delve into the nucleus, then we've got chromosomes. A section of a chromosome is called a gene, and then you can see these genes are made up of these things called base pairs, A with T and C with G. We'll worry about that more in paper 2. For now, just be aware that we've got 23 pairs of chromosomes in human body cells. Now, we talk about humans a lot because that's us. We tend to find though in exam questions, they may talk about plants and there may be 18 pairs or 8 pairs or 14 or whatever it is, but that's okay because different species have different numbers of pairs of chromosomes, okay? So if we were to put into these words in kind of scale order, we've got nucleus, then chromosome, then a section of a chromosome is a gene made of amino acids, and those are made of base pairs, okay? And this whole thing is protein, okay? Chromosomes made of protein. They're a polymer, they're a double helix. Okay. So we're talking about mitosis now, and we used to just call it mitosis. Now we say it's the cell cycle, and the second stage of the cell cycle is mitosis. We are going to need to get our spelling right on the word mitosis, okay, because it's very similar to meiosis. And this first stage is called interphase. This is where the cell goes about its normal business, doing whatever jobs it needs to do as its normal function of a cell, and the chromosomes are being copied. Okay, then we go into mitosis. The chromosomes are copied, ready to split. You can see on this picture here, they've lined up in the middle and they split through the centromere. The centromere is that middle point that holds the two chromatids together. Okay, so we can see they've split here through that centromere, and we've got one chromatid from each side of the chromosome going to each side of the cell. At the end of mitosis, we see two separate nuclei. So there's one nucleus on each side. And then the final split into two separate cells that are identical to each other and identical to the original cell is cytokinesis. We say because they've got pairs of chromosomes that they are diploid cells, think double, they're identical, and we call them daughter cells. Okay, so we've got interphase, we've got mitosis, and we've got cytokinesis. And the whole point of this is for growth and repair. This is the production of every cell in the body apart from the reproductive cells, those gametes, the egg and the sperm. Everything else is made in this way. Then we started talking about stem cells, and we said that stem cells are undifferentiated, so we're looking for cells that have not become specialised yet. And then that means that we can specialise them into whatever we need to. Okay, they can be used to treat burns and paralysis, and we can get them from three main different sources. First of all, adult stem cells, which are from the bone marrow, usually in the arm. And the good thing about these is that we can make blood cells. We still don't have enough people donating blood, so to get some cells this way that we can turn into blood cells is really useful. However, it's painful to remove those stem cells. You've got to drill into the bone. There's a risk of infection in the cut, and they only have limited uses. There's only a few different types of cells that we can get from bone marrow stem cells. Then we've got embryos. Now these embryos can only come from medical waste of IVF. IVF is when the man and the woman can't have a baby naturally, so they go to a clinic where the eggs are removed from the woman and they're fertilised in a petri dish with the sperm, and then they're put back into the uterus of the woman. Now, when she goes through this process, they will make more embryos than they need, and this is because some of those embryos won't survive. So it could be that she's had the children she wants, maybe she's had some eggs frozen and had other children, and she's had all the children she wants to have, and she's now got some spare embryos. She can have those donated to science. She can have them just destroyed, or she can donate them to science. And these embryos, only the ones that come from IVF medical waste, can be used as stem cells. Great thing about it is embryos eventually make every cell there is in the human body, so we can get lots of different types of cells. But as you can imagine, as soon as we've fertilised an egg, we are potentially really created a human life. So there are lots of ethical issues with using embryos in this way. People saying that that is a human life and we shouldn't be using those cells to make stem cells. And then we've got a new one which won't work for us because our umbilical cords are long gone, but by the time you have children, it might be that you get to store their umbilical cord. And the blood from this umbilical cord can be used to make stem cells for that person that the umbilical cord belongs to, or maybe a close relative. Okay, there won't be any rejection by the body of those cells, but there could be storage issues in the future. It will obviously cost money to have those umbilical cords stored, and it might be that you've had it stored for years and years and years, and then think, oh, can I afford to keep doing this? And then you might decide to have it destroyed, and then you might find you need it. The other issue with this is that people they're concerned really that people will kind of live more reckless lives because they'll know, oh, well, I've got my stem cells, so if I lose an arm or lose a leg, I can grow a new one using stem cells, which is technology that's in, it is happening where people are regrowing different parts of their bodies using stem cells. Okay, so 3 different sources, some positives and some negatives. They're great for our evaluate 6 markers. What if we could make an ethical embryo? An embryo that couldn't become a human life, or certainly it would if it was cloning, if it was the way Dolly the sheep was formed, but if it didn't involve a sperm and an egg. And this is what we've got, okay? So this is therapeutic cloning of an embryo. This is the bit that everybody forgets to talk about, okay? And it is the same way that Dolly the sheep was made, which we talk about in paper 2. So what we do is we take the cells that we want clones of, so we want to create our stem cells from, and we take them from the skin. So here are those skin cells. Skin cells are not very differentiated, quite simple cells, so good ones to take. Also really easy to get. Then we've got an egg cell over here, and we need that egg cell because eggs can become embryos. But you can see in that picture that the nucleus has been removed. So we've got no genetic information from the egg cell taking part in this process. And then we take the DNA from the skin cell and we fuse it with the empty egg cell, okay, and then that starts to multiply by mitosis into an embryo, and it's how we treat those cells that will dictate the kind of stem cells they will become. So whether they're going to become skin cells or muscle cells from that kind of therapeutic cloning of an embryo. Okay, so the process is therapeutic cloning. It's ethical, and it's creating stem cells that we can then program, depending on how we treat them, into whatever we want them to become. So here we've got a trachea, and this is from a donor patient. So they've died and they've said, yes, you can use my trachea. Stage 2 there is that all of the cells have been removed, so it's been cleared of all of the donor's cells, and it leaves this thing called a scaffold, which we can see in part 3. And in part 3, it's coated with stem cells from the patient who's going to receive it, from their own stem cells, less likely to be rejected by the body then. And then in 4, we can actually use that as part of the recipient's, the person who's receiving it, trachea or their bronchus. Okay. Stem cells also exist in plants. They're called meristems. Again, uses could be to clone rare species of plants, to get particular medicines from plants, that kind of thing. So we've obviously got this growing population and we need medicines for all of them. So we might need to have big supplies of these different plants with these special features, and we can use the meristems to colonize those really quickly. Next we go on to our movement of molecules. Okay, we talk about diffusion first. We know lots about diffusion. We know it's this movement from high concentration to low concentration. That happens quickest when there is a really big concentration gradient. So if there were already some cells here, there would be less of a concentration gradient, so the particles would actually diffuse slower. But because we've got no particles here at all, we see a very fast diffusion. Higher temperature, think chemistry, the particles got more energy, they move around more, collide more, and spread out more. If they've got a shorter distance to travel, then it will take less time for them to diffuse into that space. And also if there's a larger surface area. So if, for example, we've got the diffusion of a substance into a root hair cell, for example, then it's got a large surface area, so it will move across more rapidly. In this bit in the box at the bottom here, it's talking about single-celled organisms, and they've got a real thing about this, the examiners, where they talk about having a large surface area and the importance of that large surface area to volume ratio in terms of diffusion. I've done an example here on the bottom right corner where I said, okay, imagine that the cube is like a desert fox or something, and then the long oblong is like a snake. And we're looking at the surface area to volume ratio, and they do this where they get you to work out the surface area of an object, then work out the volume and compare it. And we've simplified it so the cube has a surface area of 3 to a volume of 1 ratio, whereas the snake has 4.25 to 1. So what this is saying is that for its volume, it has a larger surface area than the cube. So therefore, it's more likely to have substances diffuse a lot faster through it. And also it links in with heat loss in animals and that kind of thing as well. One to look out for. Then we've got these 4 examples of diffusion that they want us to know about. First of all, we've got alveoli in the lungs. These are the tiny air sacs. Lay them all out and it will cover a tennis court for a single person. Really, really thin, only 1 cell thick. That means that the gases can move across by diffusion very effectively. Okay, so they haven't got far to travel. Large network of capillaries, so lots of places for all of the oxygen to get into the blood and for the blood to move it away. And there needs to be moisture there because the gases do not move across as gases. They dissolve in a liquid and then move across. And a large surface area, so that there's lots of diffusion happening at once, lots of places for those gases to move back and forth. Then we've got the villi in the small intestine. Try not to confuse it with the cilia, which are found in the throat, which move all of the mucus away. Okay, so these are villi. Again, massively large surface area. All the same adaptations actually as the alveoli, except they also have these microvilli, which increase the surface area even further. Now, this increases it about 200 times more than if there were no microvilli, okay? And also we've got the mitochondria for active transport. Now, in the small intestine, we have all of this glucose. Okay, the starch has been broken down and turned into glucose, and the glucose is then small and soluble and is able to diffuse through the villi into the blood. Okay, this is how we get all the good stuff into the blood. Now, at first, that happens by diffusion because we've got a high concentration of glucose in the small intestine and a low concentration in the blood. But then we get to this equilibrium where there is the same concentration in the small intestine and in the blood. So the last bit of glucose removed from the small intestine through that villi into the blood is done by active transport. And because it's really high energy usage, it's only used once it's got to that equilibrium. So diffusion, a passive process occurring first of all, and then when that can no longer take place, active transport kicks in to get all of that glucose through into the blood. Fish gills, slightly tricky one. So we've got, fish do not breathe, okay? Fish take in water, so they open their mouth and water flows in. When they close their mouth, it forces that water over the gills. As the water flows over the gills, oxygen that's dissolved in the water diffuses into those gills, which are full of blood capillaries, okay? And it's a countercurrent flow. One thing, the water goes in one direction and the blood in the gills goes in the opposite direction. It basically enables a diffusion gradient at all points across the gill. Because when that water first hits the gill, the water is very high in oxygen, but the gill has actually, the blood going through the gills actually collected lots of oxygen from further along. So it's got a lower concentration, a higher concentration than it did further along, but still lower than the water. Okay, so if the water at this point here is, say, at 100%, the concentration of oxygen in the gill might be at 90, so we've got a diffusion gradient. As that water goes across the gill, it's losing more and more oxygen. Let's say that the oxygen is now down to about 10%, but because this is where the blood starts flowing, hasn't collected any oxygen yet, so it's probably at 0, so we still have a diffusion gradient. A countercurrent flow allows a diffusion gradient every single point along that gill. Okay. Then we've got gas exchange in leaves. We've got these guard cells work in pairs. You can see the 2 guard cells here. Stomata is the empty space in the middle. Okay, please don't ever say that stomata can get damaged. It is a hole. It is an empty space. Okay, and that is where gas exchange occurs, again, by diffusion. High concentration of carbon dioxide outside of the plant compared to inside because it all got used up in photosynthesis. Therefore, it diffuses in. Opposite with oxygen, okay? High concentration inside because of the process of photosynthesis, so therefore it diffuses out. Okay, at the same time, obviously there's going to be some water loss, and we talked about that with the transpiration through the xylem. but that is there as a byproduct. Okay, it's the main purpose is gas exchange. On a really dry day, those guard cells will close, reducing water loss. As the day becomes dry, water is lost from these, and in losing water, it causes them to flatten out and close, which is a really good defense mechanism for the plant to not lose too much water. Okay, water never enters through the stomata. Water only ever enters through the roots, and it can exit through the stomata. Okay, osmosis. Look at the definition really carefully. That is going to be worth 3 marks. Movement of water from an area of high concentration of water to low concentration of water through a partially permeable membrane. It always happens through a membrane, partially as in some stuff goes through, other stuff doesn't. And if an area is high in water, it's going to be low in sugar or low in salt or low in whatever it is that's in there. And so if something is a low sugar concentration, that's not the same as low water concentration. So we must say movement of water from a high concentration of water to a low concentration of water. We could alternatively say movement of water from a low concentration of sugar to a high concentration of sugar, but to me that doesn't make logical sense. So let's remember this sentence at the top, and that's going to give us 3 marks. Okay, and dilute, this is backing up what I said, the dilute solutions have a high concentration of water. So if we just say from high to low, that's like getting ourselves jumbled up the wrong way. Okay, so if you were a concentrated solution, it means you've got a low concentration of water. Okay. Another required practical, second one here, we do this graph. We had all these bits of potato, could be carrot, could be sweet potato, could be egg, could be anything. And we put them into these different concentrations of solution. We use sugar. This question is an exam question using salt. Okay, and looking at how much mass is gained or lost helps us to work out the concentration of whatever the solution is inside that particular food. So we've got some gain in mass. This is where the concentration of water was higher outside than inside, so it moved in. We've got some loss. This is where the concentration of water was higher inside the food than outside, so it moved out. And then where we draw our line of best fit, which is a really popular question, where it crosses and neither gains or loses mass, that will tell us the concentration inside that particular food. And that's really useful because when people are making that food packaging that goes in supermarkets of how much sugar is in a potato or whatever it might be, this is how they're establishing that. Okay, now what we did is we looked at start mass, end mass, change in mass. Then we did change in mass divided by start mass times 100. And that meant that we could see the percentage change in mass. And that's really useful because none of our bits of potato started off the same mass. So it allows us to compare the results. Remember that word compare, we use it a lot, okay? So we can compare the results fairly. If we had one bit of potato that started at 1 gram and one bit of potato that started at 10 grams, okay, the 1 gram loses 0.1 of a gram. The 10 gram loses 1 gram. You'd say, my goodness, 1 gram lost compared to 0.1 of a gram. That one is much different. But when you do it as a percentage, they've both lost 10%. So converting to percentage change in mass is a really useful way to compare results. We also dried our potato lightly on a paper towel before measuring the mass to remove any excess water. Another one that they like to talk about to make it a fair comparison. Put in here some of the key words, things that we should be aware of. Resolution, particularly important to that osmosis practical. Measuring to a large number of decimal places because the changes that occurred were very, very small. Okay, we only saw, you know, a tiny mass gain or mass loss. The independent variable, the thing that we're changing in our experiment, the dependent variable, what we're measuring, and ultimately in that osmosis practical, we were getting to percentage change in mass. That was the ultimate thing that we were eventually comparing. Controlled variables, all the things we keep the same. So for the osmosis practical, it would be things like time spent in the solution, variety of potato or whatever it might be. Okay. Once we've got all of the things right in our experiment, so we've reproduced it, which is other people reproducing our experiment, or we've repeated it if we do it ourselves, both are part of being reliable. Once we've got all of these aspects of the experiment right that allow us to appropriately investigate, we say we've got a valid method. So if you're asked to plan a valid method, you've got to have in there precision, accuracy, reliability. Accuracy, were the scales on 0 when we started? Precision, have we got a good high resolution to our equipment? Reliable, have we repeated it? Active transport, our last movement of molecules. So we've got this one, I call it the greedy one. We've already got lots of a substance, but we want that last remaining bit that's away from us. And it's movement from a low concentration to a high concentration. And we say that is against the concentration gradient. It requires lots of energy, and the energy comes from respiration, which, as we know, takes place in the mitochondria. And there's two uses of this. Mineralized in root hair cells. So once diffusion's happened, any extra minerals going into root hair cells from the soil is by active transport. And I already talked about that sugar moving from the small intestine through the villi into the blood, starts off with diffusion. After that equilibrium becomes active transport. And it uses these things called carrier proteins, and they help to move the substances, those particles, across the cell membrane. Okay, we've got our triple content now. This was the agar plates. So we've got this agar jelly, which is rich in nutrients. It's been heated to a high temperature before we use it to sterilize it and kill any bacteria. Before we use it, we then store it in the fridge to reduce any bacterial growth, just like we would store our milk. We then inoculate the plate. This means that we are going to put onto it the bacteria that we want to culture, and we're going to put on any kind of treatment. As you can see down here, these have had a plant extract put on these discs. We're also going to have some of the discs, or a disc at least, with no treatment on it at all. This is a comparison again, so that we can't say, oh, well, it's just paper that stops bacteria growing. It's actually what's on the paper that stops them growing. To inoculate the plate, to put the bacteria on, we use a wire loop that's been sterilised by heating in a Bunsen burner, and we only remove the lid when we need to. We try not to turn it upside down either. After inoculation, we put some sellotape on the agar plate to hold the lid on, but being careful to allow air gaps so that aerobic respiration can take place. And we're only allowed to store those plates at 25 degrees C, not 25 degrees because that is an angle, 25 degrees C in school, and they can do that at higher temperatures in industry because they've got safer equipment to store those in, and they get the results quicker. That bacteria will culture quicker. We then have to calculate the zone of inhibition. Those white areas on that plate are the areas where the bacteria has been killed, and the antibiotic or the plant extract or whatever it might be has diffused through the agar and killed the bacteria further away from the disc. And to work out that zone of inhibition, we use pi r squared, r being the radius, but if we measure the diameter first and then half it, we will get a much more accurate reading. So pi r squared to work out the area of the zone of inhibition. We've got an exam question here, which I won't talk through. You can have a look at on your flashcards, or alternatively, you can pause this video and have a bit of a read. I've also put in here a selection of longer exam questions taken from past papers. Again, that's something that you can work through and try and answer some of those questions yourself. And then another exam question there if you want to have a look. And here we've got some 6 markers. Okay, so these are ones that maybe you could plan to answer on some flashcards and then learn them as model answers for in the exam situation. So there was B1 in a nutshell. Ended up being a bit longer than I thought it was going to be, but hopefully you found that really useful and you can refer back to any points that you need to. Hope you found that useful. Thank you.